Why are there not more people talking about the big questions in life? Let's dive right in. We explore the most controversial ideas in religion, sex, psychology, AI, architecture, and more. Join debates, speeches, and performances from world-renowned thinkers like Richard Dawkins, Deepak Chopra, Sam Harris, and many others. Dive deep into multidisciplinary viewpoints and expand your mind with us. Subscribe to Deep World today, and let's redefine the boundaries of thought together.
My name is Dr. Michael Levin, and I direct the Allen Discovery Center at Tufts University. And I'm here to tell you how the future of medicine is going to be made possible by cooperating with cellular intelligence. Your body is built by cells. In fact, one cell, the fertilized egg, is responsible for building any of these remarkable anatomical patterns. How do the cells know what to do? Early embryonic cells work together to build this exquisite complex structure. Here you see a cross-section of the human body around the torso. The DNA specifies the proteins that every cell gets to have. But how do cell groups cooperate to build specific organs, and how do they know when to stop? And how can we induce cells to rebuild any of these structures when they are damaged by injury, disease, cancer, and so on? All of these problems have one fundamental thing in common: birth defects, traumatic injury, aging, degenerative disease, and even cancer could all be solved if we knew one thing: how to convince cells to build whatever new organs we needed. This is regenerative medicine. All of these problems could be fixed if we had control over the anatomical structures that cells build. Now, we know this is possible because some animals, such as salamanders, can restore complicated structures throughout their lifetime. So, for example, if they lose a limb, this limb will grow back. The salamanders can grow back their eyes, their jaws, their hearts, portions of their brain, and ovaries. And throughout adulthood, they restore perfect copies of the original. So we know this is possible. Now, we don't have any modern technology that can do this. If we send a complex robot into a far-off environment, such as this Mars rover, and it loses a portion of its structure, it doesn't come back. So we don't have any technology that is as good as the biology that we see around us. Here at the Allen Discovery Center at Tufts University, we are learning to understand collective decision-making by cells, and we're finding ways to control biological growth and form to develop new biomedical treatments. Now, I want to introduce you to a kind of animal called planaria, and these are really the champions of regeneration. They can be cut into many pieces, in fact hundreds of pieces, and every piece will regrow exactly what's missing, no more, no less, to give a tiny, perfect little worm. In fact, these animals are so regenerative, they've achieved immortality. There's no such thing as an old planarian, and every piece will continue to regenerate perfect, healthy new tissue. They are, in fact, so regenerative that they are teaching us something very important about the relationship between the mind and the body. It was found that planaria that are taught a particular fact, such as that food can be found in a certain spot in their environment, can then recall this fact even after their heads are amputated and grow back. So the information that a planarian picks up during its lifetime is stored throughout the body, and this information is imprinted on a brand new brain when the head regenerates. This is telling us that there's a profound relationship between the information that is learned throughout the lifetime of a creature and the body structure that forms its various organs, including its brain. What really is a memory? How is it stored, encoded, decoded, and propagated from various body tissues into the brain and vice versa? This addresses not only profound topics in the philosophy of mind, but also the biomedicine of degenerative brain disease, because at some point, human patients will have cellular therapies delivered into their brain, and we need to understand what will happen to the personality and the memory of such patients. So these model systems are helping us to understand some very basic questions about memory, personal identity, and the controls of organ structure. Biology today is focused on the molecular hardware. Many labs are getting very good at manipulating individual genes, proteins, and pathways. But what we need to do in addition to this is learn to control the software of life. Several key insights allowed computer science to drive the remarkable revolution in information technology that we see today. Back in the 40s and 50s, the way to program a computer was through physical rewiring. And you can see she's doing this right here. In order to get this machine to do something new, she would have to change the wiring. She would literally have to physically alter the structure of the machine. But what computer scientists figured out is that if the hardware is good enough, then you can control it with software, meaning you can control it with stimuli or inputs that change its behavior without having to change the structure of the machine. And this gave rise to lots of applications that we use every day that show us how powerful this concept of reprogrammable software really is. And so I want to show you a new path that we found to be able to do the same thing in biomedicine. Now, in the brain, we have some hardware which consists of cells known as neurons, and these neurons communicate with each other through channels known as electrical synapses, and they conduct electrical impulses. And this network of electrical impulses supports a kind of software. So here you can see the electrical activity of a zebrafish brain while this zebrafish is thinking about whatever it is that fish normally think about. And it's possible now to begin to decode the information content or the thoughts that are driven by all of these sets of electrical activity. Now, what we found out is that, in fact, all cells, not just neurons, have this ability. All cells drive electrical signals. All cells communicate with each other through various kinds of electrical connections, and it turns out that this ability to store and process information in electrical networks is universal. And what you see here in the bottom right is an early frog embryo, and all the different colors are showing you in time-lapse video all the electrical conversations that cells are having with each other to determine which cells will make the right, left, top, bottom, anterior, and posterior parts of the developing tadpole. And these bioelectrical patterns are an extremely important instructive influence over what happens in embryogenesis. So these patterns are what we call bioelectric scaffolds or bioelectric pre-patterns. Here you see another time-lapse of a frog embryo putting its face together. And so here's one frame from that video, and you can see the white regions are cells which have a very particular bioelectrical property. They're depolarized, and you can already see in this new tissue, you can already see the outlines of the frog face. There's the eye, the mouth, the organs on the sides called placodes. You see all of this that's going to be formed later on. So this is an instructive pre-pattern, and if this pre-pattern is disrupted, then the gene expression will change, and then the anatomy will change. And in fact, we have examples of pathological changes. Down here you see a tadpole that bears a human oncogene, which is going to induce a tumor. But even before that tumor becomes obvious, you can detect it non-invasively using a fluorescent voltage-sensitive dye, which reveals that these cells are going to have an aberrant bioelectrical potential, and they are not going to cooperate with the rest of the body. They're in effect shutting off the electrical connections with the rest of the tissue and going off on their own. They're going to treat the rest of the animal as just environment, and they're going to revert to a kind of unicellular ancient existence, and this is what we see in metastasis and cancer progression. So watching these bioelectrical signals is really important, not only for understanding where developmental anatomy comes from, but also as potential diagnostics for birth defects and also for cancer. Now, what we've developed are ways to control these electrical properties in cells using pharmacology and light. So we use drugs to open and close these ion channels that are proteins that produce electrical signaling in every cell, and we can use these drugs together with light stimulation to provide various kinds of electrical pre-patterns directly to growing tissues. And so one application of this here is in the normalization or reprogramming of tumors. Here you see a tadpole embryo that has been injected with an oncogene, and normally it forms a tumor, and you can see that here with these labeled in red. But even though that oncogene is strongly present, you can suppress the appearance of the tumor by co-injecting with the oncogene a specific RNA encoding an ion channel that normalizes the electrical properties of those cells. So by controlling the electrical properties, even though the genetic mutation is present, you can suppress the appearance of the tumor. You can force those cells to cooperate with their neighbors to remain in electrical connection, and by enforcing the correct bioelectrical state, you get normal tissue instead of a tumor. We've also used the same technology to repair brain structure in tadpoles that was damaged by mutations or harmful teratogenic drugs. So here is what a normal tadpole brain looks like. A truncated, misshapen brain results from mutations in genes like Notch or in exposure to alcohol, nicotine, things like that. And what we found is that by manipulating the native ion channels in a very particular way using drugs, in fact drugs that are already approved for human use, we can restore the bioelectric pattern that is appropriate for telling these tissues how big and what shape a brain they need to have. And in fact, then the correct brain is built. And these tadpoles have not only the normal shape of brain, but they have normal gene expression, and they even have normal IQs. So we now have a computational system, a set of programs that allow us to find the correct drugs to induce an appropriate bioelectric state to repair some very drastic craniofacial defects. And in fact, what we also discovered is that tissues have anatomical memory. So here's a planarian, and normally when you cut a planarian into pieces, every piece gives rise to a normal flatworm, one head, one tail. Now we can perturb briefly this bioelectric circuit after cutting and confuse the piece about how many heads it's supposed to have. In fact, we can reset that bioelectric pattern to dictate having 2 heads. And in fact, the middle piece here has grown, you can see that here, has grown 2 heads. But the most amazing thing is that if you then again take the middle piece out of that worm and culture it again in plain water with no more manipulation of any kind and simply let it regenerate, it will once again make a 2-headed animal, despite the fact that it has a normal genome, a normal genomic sequence that we have not edited. And so in perpetuity, as far as we can tell forever, every time you cut these worms, you get a 2-headed animal. So think about what this means. The pattern to which these cells build after damage can be rewritten, can be bioelectrically respecified towards 2 heads, and in fact now we can set it back to normal as well, in a way that only changes the information stored in this electrical circuit. The DNA is completely normal. We have not changed the genomic sequence of this animal. So we have now found that some information is present in the physiological circuit that is not directly in the genome, and this physiological circuit can store information that is changeable by us. In fact, not only can you ask these worms to make a second head, if you perturb the bioelectric network, you can also get them to build heads that are appropriate to other species of worms. So here you see a normal Dugisia doratocephala with a nice triangular head. By disrupting that bioelectrical gradient, you can make their fragments regenerate flat heads like a Piphalina or round heads like an S. mediterranea. And not only the external shape of the head changes, but also here in green that you can see the shape of the brain becomes like these other species, and the distribution of their stem cells, which you can see with the red dots, are also changed to be just like these other species. And again, this is done without changing the genomic sequence. So extremely important information is present in this bioelectrical network that guides cell activity towards building specific different kinds of structures. A very important thing to understand is the concept of nested cognition. At all levels of organization in biology, from molecular networks to subcellular structures to neural networks to whole tissues, organs, whole organisms, and even groups like ant colonies, there is the ability to solve problems at every scale. Every one of these levels of organization has a degree of intelligence that allows it to solve various challenges and changes in their environments and maintain optimal function. If we understand the scaling of this ability, how it is that the activity of the simpler levels underneath give rise to intelligent activity at higher levels, we will achieve transformative applications in medicine and artificial intelligence. The idea is not simply to understand the mechanisms by which these things happen, but to understand the information, how every layer of the system is able to produce more complex, intelligent responses and robustness at higher levels. This will not only allow us to have much better control over organogenesis for regenerative medicine, but also we can use this kind of information to develop new ways of achieving artificial intelligence. In summary, all cells communicate electrically to form networks that make decisions about growth and form. By reprogramming this physiological software, we can unlock the promises of regenerative medicine. We can go beyond genomic editing of the hardware of life to provide definitive solutions to injury, aging, and cancer by learning to convince cells to build whatever we want them to build. I want to thank the postdocs, collaborators, and students that have contributed to the work that I've shown you today. And of course, I want to thank my funders who support our research. And I thank you for listening.
Thank you for watching Deep World. Stay tuned for more explorations into ideas, arts, and sciences. Don't forget to like, share, and subscribe. But before you go, let's take a moment to explore the power of curiosity. Imagine a world where questioning the status quo is not just encouraged, but celebrated. Where every why leads to a new adventure, a new discovery, and a new way of thinking. This is the essence of Deep World. Curiosity is the engine that drives innovation, creativity, and understanding. It's what pushes us to look beyond the surface, to dig deeper, and to seek out the truths that lie beneath. It's the spark that ignites the fire of knowledge and the fuel that keeps it burning. So whether you're pondering the mysteries of the universe, the complexities of the human mind, or the beauty of artistic expression, remember that curiosity is your greatest ally. Keep asking questions, keep seeking answers, and never stop exploring. Join us on this journey of discovery, and let's unlock the endless possibilities that curiosity brings. Stay curious, stay inspired, and stay tuned to Deep World. Until next time, keep diving deep.